The next talk is by Sunil Shetty. He is a PI at ATREC, Navi Mumbai. Sunil did his PhD with Professor Umesh Varshney at IISc, and then he went to University of Basel with EMBO Fellowship. He also won the INSA Young Scientist Award, and now he's looking at ribosome heterogeneity and stress response.
Yes. Thank you. I think I'm audible. Why? Sit. Okay. Hello all. So I recently started my group in ATREC. So if somebody doesn't know where ATREC is, we are in between Pune and Mumbai, and we are close to Mumbai, but we don't have that crowd of Mumbai or the hotspot. So it's a bit pleasant place, ATREC. So and my group works with two model systems. One is yeast and another one is liver cancer. So these are the two systems, and we study regulation protein synthesis and mainly in the context of ribosome composition and how they play important role in the like the translation regulation. So to this audience, I don't have to explain about the ribosomes. So ribosomes as such, all the like compositions are known, the structures are known. But the concept about the ribosome heterogeneity mainly derives from the variation in the ribosome content. So the ribosome content can be different with respect to their RNA sequence from bacteria. Like even if you take simple E. coli, it has a minimum 7 copies of rRNA sequence, and they do have some variation in the sequence. Similarly, but if you go to eukaryotes, it becomes hundreds of copies, and in cancer also there are known that subset of RNA expression is changing. So how those RNA sequence affects the ribosome function, we don't know. There are RNA modifications which can also be altered. There are ribosome stoichiometry, means not all the ribosomes can have the same composition. They can have a different, like some ribosomal proteins might be missing, or the paralogs, where there are multiple copies of the, like different copies of the same gene can exist, and it can be different in that terms, or the ribosomal protein can get modified, and they might can also now function differently, or there may be the associated factors which we call ribosome associated factors. So my talk will be mainly focusing on the ribosome associated factors as well as the ribosomal protein paralogs. So these are the two components we are visualizing in this talk. So as I said, there is a debate. Like even if I'm happy, like that, today there is no Rachel Green, because she says that this concept of heterogeneity may not be true. It's just a number basis. Like the translation regulation is mainly based on the abundance, not about this like the specialization is what they claim. But yes, it's an open, a little bit debatable aspect. But what we are trying to say that even normal cells can have such subpopulation of the ribosomes, but our window of looking at is like we use a stress as a condition and ask question, any of these factors change during that particular stress, and now can we investigate that mechanism more in depth. So that was the purpose. That's why we look into the stress conditions, and mainly with the window of a kinase called TOR, target of rapamycin. So this kinase has been considered as the master regulator in eukaryotes, because it can sense multiple stress conditions and modulate the protein synthesis. So this there are two complexes in the by this kinases, TOR complex 1 and TOR complex 2, which is conserved from yeast to humans. And TOR complex 1 can integrate multiple stress conditions, like it can sense multiple stress conditions and then suppress, like in that context, it can then, if it is inhibited, it can lead to the decrease in the translation and ribosome biogenesis. And to do that, we can just simply add rapamycin and we can inhibit the translation. So if you, like the polysome profile, everyone must know now, but please, again, I will reiterate that we mainly like look in the view of the polysome profile, where in the normal condition, we have this ATS population and the polysomes. But if you treat the cell with rapamycin, what we see is that decrease in these polysomal fractions, but increase in the accumulation of these ATS ribosomes. And this is one of the indication that the translation is gone down in these cells. And this population, what we say ATS, can be either it can be a monosome, where the single like the one ribosome translating one mRNA, occupying one mRNA, or the inactive ribosomes. So to begin with our study, what we had done was analyzing the ribosome composition by doing the proteomics of these different fractions. And one of the protein, this is the one of the protein STM1 is what we focused on, because it was already known factor known to bind to ribosome. And what it was known that it can trap ATS complex as a dormant ribosome. So this is the 60th and 40th region. So this protein occupies near the mRNA tunnel and can trap those ATS complexes. And basically, as per this theory, it should have been a inactive ribosome, which is not functional. But when even in the mass spec and as well as when you do the Western blot for the fractions, what do we see that this STM1 is also present even in the polysomes. That is like these are the population which are supposed to be actively translating, but we do see them as in the these fractions. And when you treat the cells with the rapamycin, they now more abundant in the ATS population. So that was contradicting with the known aspect, because if STM1 is there, then it cannot occupy the mRNA. So, but then what we realized that it's mainly because of the effect of the sucrose gradient, where we were using the 50 millimolar KCl. And if you now slightly increase the concentration of the sucrose density, the same lysate if you run, what we see is that we can, the STM1 is not appearing in the polysomes, but it's mainly in the free region. And very small proportion in the active condition is present in the ATS population. And but upon rapamycin, you see this accumulation in the ATS population, saying that, okay, now it makes more sense that upon translation inhibition, maybe this ATS complex stabilized by the STM1 is accumulated. Further, we had actually shown that if you don't have STM1 in normal condition, as I said, wild type cells form the ATS complex upon TORC1 inhibition. But if you now don't have STM1, like if you delete STM1 in yeast, what we see that instead of ATS upon TORC1 inhibition, you accumulate the free subunits, saying that indeed this dormancy factor was actually stabilizing the ATS complex under starvation conditions. So further, we had actually shown this was my previous postdoc work, where this STM1 factor is directly phosphorylated by TOR kinase, and there are 3 phosphosites, serines, which are getting phosphorylated by the TORC1. And further, these phosphorylations are indeed having an important role, as in the, if you bring the a mutant which cannot be phosphorylated, that STM1 can bring back the ATS complex. But if those are phosphomimetic mutants, where the serine is mutated to glutamic acid, these mutants cannot bring back the ATS population, saying that these phosphorylations are indeed inhibiting the ATS complex formation by the STM1. So based on this, what we had proposed the model that TORC1 in the normal condition where the TORC1 is active, this STM1 gets phosphorylated and is kept away from the ribosomes, so that it doesn't form the dormant ribosome. But upon rapamycin treatment, these phosphorylations are gone, and then you form this ATS complex. But now in the lab, the Pooja, she joined as a PhD student, and she again, we showed that under low salt concentration, this STM1 was binding to the polysomes, but at high salt concentration, this interaction was not there, saying that maybe the sucrose density gradient salt concentration can impact our observation about the ribosome associated proteins. So because our previous mass spec was done with the 50 millimolar gradient condition, she repeated this again the the proteomic analysis, but now this time she used the 150 millimolar KCl gradient, and then again collecting the fractions and doing the mass spec of these fractions and trying to identify what all the stable ribosome associated factors are there. So what we see, this is nice, these are the expression of the proteins, ribosomal proteins, as you expect from the polysome fractions. Upon TORC1 inhibition, these polysome signals, the ribosome proteins in the polysomes, are now accumulated in the ATS fraction, suggesting that indeed our mass spec analysis can agree with what polysome profiles look like. So the ATS accumulation we see in the proteomics. And then Based on this, she looked for the non-ribosome, like ribosome associated factors, which are non-ribosomal proteins. And we do see several proteins like here, which are co-migrating with the polysomes. Those are now gone down under rapamycin condition, but many of them also accumulate in the ATS fraction, saying that their interaction with the ribosome may not have perturbed under a torque1 condition, but they are still a novel ribosome interacting proteins, which we don't know yet what it is doing. And we also have some interesting candidates where these factors are now more associated under torque1 inhibited condition. Whether these factors help in the translation regulation under starvation condition is also open question. So these factors she is characterizing now. But apart from this, what she also focused was looking at the ribosome composition itself, like ribosomal protein content itself. What we see that there are lots of ribosomal protein paralogs. Like in case of yeast, because yeast has undergone a genome duplication, even in the haploid cell, we have almost many of the ribosomal proteins, like the RPS, out of 33, there are 24 paralogs, 24 genes which have a paralogs. In case of RPL, 33 genes have a paralogs. That means they are duplicated. So she was looking at the behavior of these paralogs, whether do they behave similarly to the rapamycin treatment. What she was observed that one of the paralog, which is RPS 9A and 9B, 9A was not accumulating in the, like this is the quantification of the only the ATS fraction. She is comparing the ATS fraction of the normal condition and the torque1 inhibited condition. Ideally, theoretically, we expect that ATS, all the ribosomal protein in the ATS should go up upon rapamycin, which is we see for the RPS 9B, but not for the RPS 9A. So, so then she asked a question whether do these paralogs behave differently under different conditions. That was the question we were interested. So for that, if you look at focus on the RPS 9A and 9B, they're quite similar, both of them. There are only 3 amino acid changes. One, this K to R change in the 40 position. And there are 2 glutamic acids, which are higher in the RPS 9A. So the RPS 9A, which is less abundant protein. So that has a C-terminal tail, which is a bit longer than the RPS 9B. And this protein is supposed to be occupying this region, entrance tunnel of the mRNA. And what we could see that with the help of Varun from JNCS, that this region, the C-terminal region of the RPS 9A, might be potentially having some interaction with the EF3 complex component. So we still don't know whether it affects the initiation factor or something, this C-terminal region. So what we have done is to delete one of the paralogs. So we have generated the strain where either RPS 9A is deleted or 9B is deleted. 9A deletion do not show any phenotype so much, like so far we could not make any phenotype. But 9B deletion, we can see that it is growing poorly even in normal condition, but it becomes more sensitive upon a rapamycin treated condition, saying that like the 9B has a better function. But also then the expression question was there. So we flag tag those paralogs and then check their expression. We were expecting that 9A will be very low abundant, but it doesn't look like, at least the expression wise, they are like at least 1/3 of the 9B. So, so it's interesting that why 9A deletion doesn't have so much phenotype, but 9B gives a major phenotype. When we looked at the polysome profile, what we see is that 9A deletion doesn't hamper the polysome profile. It's quite similar to the wild type. But 9B, if we delete, it gives a ribosome biogenesis defect, like the 40S particles are not made properly. So you end up in accumulating the 60S and you have quite a low amount of ATS and the polysomes. But these cells are surviving. It's not that they're completely dead. But yes, you can say that 40S biogenesis is affected. So 9B, the particles which don't have 9B maybe are degraded faster. So only certain 40S particles which contains 9A are now doing the job of the translation. So this was a nice system for us. So we had this phenotype like the polysome profile where we can see the biogenesis defect. So then we asked the question, can we now bring back, if we bring back 9A and 9B separately in this strain, will we be able to rescue it? So as I said, 9B deletion strain is rapamycin sensitive. When we bring back either 9A copy or 9B, so in this strain, they should have only a ribosome which contains 9A and there is no 9B. So in this strain, also you see that a rescue of the growth defect, saying that any one of the paralog can do the job. Then the next question was how the polysomes looks, because of, we said that there is a biogenesis defect. What we see that if you, in this delta 9B, if you bring back the wild type 9B, you see that nicely a rescue is there. And upon rapamycin treatment also, you can see that they can form the ATS complex. But in case of 9A strain, which contains only 9A, when you treat the cells with the rapamycin, we do not accumulate this ATS peak so much. And even the polysome shutdown is very less. So this is interesting that a cell which contains only 9A, maybe the dormant ribosome is not happening, like forming, the STM1 may not be forming. That was our hypothesis, maybe because there is no ATS accumulation happening here. So that's why we check the STM1 occupancy in this ribosome. So in delta S9B, which contains only 9A paralog, we did not see the STM1 accumulation. So that's what a nice thing for us. We thought, oh, our hypothesis might be working, that S9 here, this S9A containing ribosomes are not showing the accumulation of the STM1 upon rapamycin treatment. But wild type did show the accumulation, but also 9A containing do show some accumulation. So we are still figuring out why 9A is, like the polysome is not, like the ATS is not accumulated so much upon rapamycin treatment, but we still see STM1 accumulation. So with this, the part which I didn't tell you that there are multiple forms of dormancy factors in any given system, like from bacteria to eukaryotic system. So in yeast, there are two dormancy factors are known. One is STM1 and other one is the LSO2, this factor. So now we are in pondering whether there is a difference in the like dormancy, like whether subset of ribosome behave differently under stress condition. That's our hypothesis. We still have to delineate whether LSO2, what is the occupancy of LSO2 and all. So this is one still half-cooked story we have in the lab. Second, another half-cooked story in the lab is about the phenotype we stumbled upon. Sorry, yeah, please. Yeah.
So the one that shows rescue, but
Right.
it doesn't go through STM.
No.
Does it also rescue the biogenesis defect? Because
Yes.
that could be the problem that.
No, that's what. So here, the 9A complemented one, it rescues the biogenesis defect. Here, compared to this strain, 9B delta, we don't see that 60S peak now. So the biogenesis defect is rescued by the 9A.
Okay.
But the dormancy may not be is what we think, but we don't know still whether this population is resistant to translation repression or whether some other dormancy factor is binding. We don't know actually.
Okay, so maybe I'm
more
oriented now. Thank you.
Okay. Yeah, I'll go to second part. So second part where we had was it's another observation, like it's about the TIF3. TIF3 in yeast is like a homologue in case of mammalian system. It is called EF4B. So EF4B is a factor known to promote the activity of EF4A. EF4A is the helicase which is supposed to help in the translation initiation. So what we had observed that if we overexpress TIF3 in yeast, what we see that it can give resistance to a drug called myriocin. So that observation is interesting. So what is myriocin? So myriocin is a blocker of sphingolipid synthesis. So in yeast, there are, like in all the eukaryotes, you have these sphingolipids, and this yeast also has a special category of sphingolipids, and both TORC1 and TORC2 pathways are known to regulate the synthesis of these sphingolipids. Sphingolipids can form the they will form ceramides, and then they will form the complex sphingolipids, which are important for the various functions like formation of the lipid rafts and which are involved in the signaling, regulation of signaling pathways, as well as mitochondrial membrane function and all. So they do have essential function. And what we see that if this myriocin is a drug which blocks the function of LCB, which is the first rate limiting step of synthesizing this sphingosine. So as I said, the TOC2 branch where TOC2 activates the YPK2, which is well known that YPK2 gives resistance to the this myrcene drug, because it can now increase the synthesis of sphingolipids and thus block overcome the inhibition. But as I said, in literature, what is also known that TORC1 is regulating the function of the 4B. So that's why what we asked that question, since 4B is giving the resistance to the myrcene, so whether since 4B is known to activate the EF4A and that will lead to the translation along the translation initiation, we asked whether TIF1 overexpression, TIF1 is the EF4A in yeast. So if we overexpress TIF1, can we also get the same phenotype? That was the first question we had in mind. But interestingly not. So it's not the EFA overexpression leads to this resistance, but only TIF3 overexpression can give rise to the resistance to this drug. And this was the positive control, as I said, downstream of TORC2. You are supposed to get, like, if you overexpress a catalytically active mutant of YPK2, it is known to give the resistance to myrcene. So that's nice. So, but then we also observed that TIF3 overexpression can give resistance to rapamycin. So, saying that both downstream of TORC2 and TORC1, maybe TIF3 is working. So, still we need more data. But what main concern here was, what if this is like something completely specific to drug, maybe some random impact of the drug resistance by TIF3 overexpression, something else is happening. So then that's why what we did was perturbing the enzyme activity of the LCB1. LCB1, this is a trimer complex which is involved in the sphingosine synthesis, and these all three are essential genes. So we cannot delete them. What we could do was, using the AID system, tag them with the AID, which is auxin indisible degrowth system. And we can, using the auxin, we can deplete these LCB levels. So we tag the LCB1 with the AID tag and use auxin to deplete the activity of this function. And indeed, if you deplete the treat with the auxin, these cells will grow poorly. So even in this genetic system, what we could see that if you overexpress TIF3, you can rescue the growth defect, saying that maybe downstream of LCB1 activity, this TIF3 is doing some positive function. That's why we are able to get this rescue. So then the obvious question was whether is it via translation. So when we treated the cells with myrcene, what we can see that indeed translation goes down by myrcene, and it is kind of expected because myrcene is since blocking the sphingolipids, and sphingolipids are important for the signaling hub. It can be indirect impact by the this drug. But as I said, this translation goes down. But TIF3 overexpression context, the translation status is very confusing. I'm still not able to conclude anything. Like, it shows a little bit resistance, translate repression, but not conclusive. So we are trying to do more experiments with this. But what we could see that if you delete TIF3, the cells are much highly sensitive to myrcene, and in TIF3 knockout cells, we see already a very low translation. Those cells are sick, and there is very less translation, and there is instead there is an accumulation of the ATS population. And treating those with the myrcene, there is no much impact as expected because the translation is already low there. So with this, what other genetic observation we could see, because as I said, both TORC1 and TORC2 have an impact on the sphingolipid synthesis. We did another experiment where, as I said, YPK2 is a downstream target of TORC2 and already known regulator of the sphingolipid synthesis. In wild type, as I said before also, that YPK2 overexpression can rescue the myrcene, like the sensitivity. But if you now have a delta TIF3 strain where the cells cannot, like they're sensitive to myrcene, but if you now, in this background, if you overexpress the YPK2, which is the upstream regulator, we do not rescue this. The resistance is not coming back. Saying that somehow YPK2 downstream downstream of the YPK2, TIF3 is involved to give resistance to the myrcene. So most likely TIF3 might be involved in the sphingolipid synthesis. So we are now thinking in the direction that whether the translation of these downstream mRNAs, whether they are affected by the TIF3. So the hypothesis is that maybe downstream of YPK2, TIF3 may be required to promote the translation of the sphingolipid synthesis mRNA. That's the hypothesis. Still, yeah, we have to explore the mechanism more. Yes, with this, I would like to thank the organizer for this opportunity. And yeah, we are still. Yeah, 4B. 4B is not a multi, it's a single protein. Multi-domain, you mean to say. Okay, sorry. Yeah. Yeah, good question. Like you have to tell that Prithviraj fellow. Yes, we are doing that, deleting the different domains and asking question, which domain is actually important. Yeah, that's on the way. Yes, excellent. Yes. Yes, sir. That's the hypothesis. Definitely, we believe that. So we have to show it. That's the problem. So far, evidence for the difficult, like there are known evidence for regarding the like the 3' mutate structures. Like Maria Berner's group proposed like L32 or 38 containing protein, like the ribosome, are specifically translating the certain mRNA, like Hox mRNAs, and they have depending on the 5' mutate structures. So those kind of there, but yeah, still more and more like very specific context dependent is not known, I would say. Yes.
Yeah. Nice talk, Sunil. So with respect to 9A and 9B,
yeah,
what is the similarity?
Similar.
They are parallel.
They are almost parallels. There, 99B are parallels, and there's only 3 amino acid changes. Nothing else. Only 3 amino acid change. Huh? Means 3 amino acid means 2 or less. Yeah, agree.
So
correct.
So,
yeah.
So if it is so, what is intriguing is that when you rescue it, why is it that one is not able to rescue the
other, right? Yeah, that's what. So the one aspect we had was about the abundance. Maybe the expression of those paralogs are very different, and that's why affecting the function. But for that, what the experiment now we are doing is whatever here the complementation we are doing, right? In the 9B construct, we are trying to make it as a 9A, so that promoter and everything is same of 9B, and it will produce a 9A function. And then we want to ask whether this sequence, like 3 amino acids, whether will they play any role. So once that answer, then only I can answer this question. No, they are not they are different chromosomes. Correct. Yes, those are regulations are completely different. Yes. Yes. Yeah, that's what. Just last 2 already we have done, and that doesn't give like it shows like a 9B. Okay, so there is no difference. So that's why we want to make all the 3 mutations and then conclude it. So
in the structure, do you have the complete tail?
No, that's why this structure is only partially like this blue region. What we're showing, it is from the alpha fold. This light blue region. This much is missing. Only till the dark blue.
Near 3B.
So here this much is only this blue part. Remaining one we have to. So that's a 22 angstrom is good enough distance to cover that loop region. Yeah. Huh?
No, it looks like local network will be important.
What about the others? Because you reported like I don't know how many.
Yes.
I know that some paralogs are identical in sequence, like RPS0, for example.
True.
It differs only by UTRs. So what about the others?
So there are mainly like that's what we also made the analysis of different paralogs, and there are only 5 to 6 paralogs which are sequence-wise like I would say drastically different. You can say. Otherwise, most of them are almost similar. So like only serine to threonine kind of changes they have. One of the paralog, I think it's S11.
So you saw differences in the paralog occupation of the ribosomes under rapamycin treatment with
not all, but multiple. There are 7-8 genes which are showing this difference behavior.
So why do you think this is kind of like a general mechanism that they not just S9AB but also the other alternate So it's like a switch almost, if you see that, if
Correct.
I understand it correctly.
So what we think is these particles might be having a like condition to condition, they might have a different translation activity. So what is already reported that certain paralogs are now highly expressed under heat stress or some other stress condition. So it's quite possible that which are like the S11, which shows like at least 20% change in the amino acid sequence, they might actually give like a two different type of ribosomes and behaving differently. So that will be nice. Like even like there, we didn't pick up that candidate because I think it was showing the similar accumulation. So, but yes, it can be for different stress. It can be a good candidate there to explore. But yeah,
Does TIF3 bind to this antibiotic,
which one?
for which it shows in resistance?
I didn't get which.
So the antibiotic that you studied, that's, you know,
Erythromycin? No, it doesn't.
which one? The other story.
Rapamycin,
Where is-
myrosin. Whether myrosin binds to the ribosome.
No, TIF3.
TIF3. That's what I don't. That's to rule out that part. We did this LCB knockdown because that was one hypothesis. Maybe TIF3 is sequestering the like binding to the myrosin and titrating out. Right. That was one hypothesis possible. That's why we did this the myrosin LCB knockdown strain, where even if you deplete the enzyme, you can rescue the function. Here, TIF3 overexpression. Here there is no myrosin. We are only blocking this LCB1 by depletion, and then still it's rescuing, saying that it is may not be the direct interaction or something mattering, something else is going on downstream. We have to figure it out. Yes. Yes. Translating, right? That's a good question. So we still don't know that. It's just that we can make a wild guess based on the how much of STM1 is abundant, like present in that ATS, right? Like here I showed that here, like upon rapamycin treatment, there is accumulation of ATS, like STM1, right? So based on that, we can make some guess, okay, how much ATS is accumulating here and how much STM1 is accumulating here. But very crude, like we don't know, because it's known that under stress condition, the ribosome can also undergo a monosome mediated translation. Like they don't have to go for the polysomes. They're like neuronal cells and all. They just undergo monosome translation and give enough sufficient proteins. So that way, it's still by splitting them, right? Yes, we have done that in the 450 millimolar KCl. We could split these dormant ribosomes. Okay, there I would say that still something 50%. I would say that still monosomes are the ATS are there. I can't say that completely gone. So
STM1 may be binding to a different site in low salt.
in this one, right here.
It may actually be at a sort of a position from where it can go into the mRNA channel.
Agree. So that's one favorite hypothesis we had, that in the cells, maybe it is still in association with the ribosomes, but upon stress condition, maybe it just flips into the mRNA channel and blocks those ribosomes after the translation damage. That's a favorite hypothesis we have, but we don't have evidence. So, but that's quite possible.
By some sort of cross-linking or something.
Correct. So we thought we will try to deplete the termination factors. Like if you block that translation termination splitting, whether this STM1 occupancy will it be perturbed or something. That was the experiment we were thinking. But yes, still whether because there was some hypothesis that maybe the free subunits are now trapped by the STM1, or whether the terminating ribosomes are sequestered into this. That is still a little bit debatable question. Yes.
Let us thanks.
Thank you very much.